Okay, I want to thank everybody here who stuck around to the very end. And I want to thank the organizers of the meeting for giving me the opportunity to talk about some of my research here today. So I'm going to shift gears from the previous talks and really discuss diagnostics and talk about how we're using peptide-based assays to try to improve the laboratory diagnosis of Lyme disease. Let's see, how does this? Okay, just to start off, the standard conflict of interest statement. I am an employee of Biopeptides Corp., which is a faculty-run startup at New York Medical College. And the peptides I'm going to discuss today are patented by Biopeptides Corp. And may serve as a future source of research for the company, though I won't see anything out of it. So before I get to the diagnostics we're trying to develop, I'm going to go into a little bit of background on Lyme disease itself and the problems with current diagnosis of the disease. So Lyme disease is the most prevalent vector-borne infection in North America and Europe. It can produce a progressive illness with an array of clinical manifestations, and it is in the differential diagnosis of multiple neurological and rheumatological conditions. It's a zoonotic infection caused by species of Borrelia burgdorferi sensu lato, which is a complex of 12 Borrelia species that cause Lyme disease. The three most clinically relevant are Borrelia burgdorferi, also referred to as sensu stricto, which is the causative agent of Lyme disease, predominant causative agent of Lyme disease in the United States, and Borrelia fzelii and guerinii, which combined are the primary causative agents of disease in Europe. They are spirochetes, which are highly motile corkscrew-shaped bacteria, which you can see over here. This was a time-lapse video that decided not to move, but that are present in mouse skin. They are transmitted by the bite of infected Ixodes ticks, predominantly Ixodes scapularis, but also Ixodes pacificus on the West Coast. And the reservoir hosts are small mammals and birds, primarily mice, or the white-footed mouse. The prime time for Lyme disease transmission is the late spring, early summer, and fall, and that's primarily due to the lifestyle of the tick. The tick hatches from eggs, generally in the mid-spring, releasing larvae. Now, thankfully, the larvae themselves are not infected because there is no vertical transmission in the tick vector, and they will take a blood meal on small mammals and birds and then become infected. They'll molt, and in the following spring, they will be nymphs, and the nymphs are capable of transmitting disease to humans. The primary risk of human infection is greatest during this nymphal feeding period, primarily because the nymphs are tiny little organisms. They're about the size of poppy seeds, which you can see down here in this public service advisory from the CDC. Actually, on the top of this muffin, 5 of these dots are nymphal ticks. So you can think about that the next time you're picking out breakfast tomorrow. And because of the fact that during these warmer months, people are running around in the woods in shorts and short sleeves. During this time frame, the nymphs will take their blood meal, and then they will molt to form adults. And then in the fall, the adults will have another feeding period. This is another burst of Lyme disease here, although it tends to be less than the nymphal period because the adults are larger and more visible, so therefore they can be removed before they transmit the disease, and also because at this time of the year, people tend to be more well-dressed in pants and shirts and with less exposed skin. Now, despite this, you can actually get Lyme disease at any point during the year. So you should always be doing tick checks if you've been out in the woods. The geographical distribution of Lyme disease is primarily the northeastern United States and the upper Midwest, with some cases in the Northern California Bay Area region. That's primarily due to the presence of the ticks in these sites, although, as you can see here, from 2001 to 2017, there is a great increase in the geographical distribution of the ticks, and the disease has now moved up as far north as Canada, as far south as Virginia, throughout the Great Lakes region, and is expanding on the West Coast, primarily due to climate change, we believe. Although I'm not supposed to say that anymore. It's a bad word. Along with the geographical increase, or the geographical spread of the disease, we're seeing an increased incidence, which you would naturally expect. So from 1997, when Lyme disease first became reportable, to 2017, there's been a sharp increase in the number of cases. And in 2017, there were a total of 42,000 cases that were reported to the CDC. However, the estimate is that this is only about 10% of the actual total cases, and that the rate of Lyme disease in the United States is about 300,000 new cases a year. It is also the 6th most commonly reported disease to the CDC in the US currently. So transmission occurs when the tick bites a non-infected host and begins a blood meal. The bacteria are present in the midgut of the tick. When the blood meal begins, the bacteria undergoes a transcriptional change and changes the surface proteins, downregulating a protein known as OspA, which is important for adherence in the midgut of the tick, and upregulating a protein called OspC, outer surface protein C, which is required for transmission to the mammal. Now, it's important to note that the bacteria undergo this transcriptional change in surface proteins as they're being transferred into the host, and then this changes progressively over the course of the disease, and this change in surface proteins is important diagnostically, because if you're not looking for the right antigens or responses to the right antigens at the right time, you're going to miss the presence of the bacteria. After the bacteria moves to the salivary glands of the tick and into the skin, it will begin to disseminate, first through the skin, and then eventually entering the lymph and the bloodstream. When this typically occurs, the majority of people are going to have the formation of an annular rash referred to as the erythema chronica migrans, or erythema migrans skin lesion. This is the only distinctive clinical feature of early Lyme disease, and typically has this targetoid appearance where you have central redness, a clearing, and then an outer leading edge of redness. It's believed to occur in approximately 80% of early Lyme disease cases, but of course this is not all Lyme disease cases. And when you're talking about approximately 300,000 cases a year, that means 60,000 people are not having erythema migrans. It can develop anywhere from 3 to 30 days after the tick bite, most commonly 7 to 14 days. And is so characteristic that in endemic regions, when a person is presenting to a physician with a clearly definable erythema migrans lesion, it is considered diagnostic and proof for treatment. However, not all erythema migrans are the same. There's a high rate of atypical erythema migrans. The rate as high as 30% in some reports, where you don't have that typical targetoid lesion and you have some variability in appearance. So therefore, an erythema migrans can be confused with other rashes that may present, such as bug bites from other things. Spider bites are a common thing that's misconstrued as an erythema migrans, drug reactions, and even cellulitis. I personally have known several people who've been told that they have cellulitis when they in fact turned out to have Lyme disease. In addition, the EM can form in areas that are not readily visible, such as EMs between the toes, which we've seen in the hairline in the center of the back, where it's not readily visible to an individual. And it's also fleeting and can be gone by the time a patient feels ill and seeks medical care. All of this complicates diagnosis of Lyme disease. In the absence of the erythema migrans lesion, the remaining clinical manifestations of the disease are vague constitutional complaints and signs, such as fatigue, low-grade fever, headache, myalgia, arthralgia, and neck stiffness. These are some of the more common features of early disease, considered general viral-like disease symptoms. It's complicating trying to diagnose someone with these symptoms because there's a high incidence of these symptoms in the general population. There's a high incidence of fatigue, depending upon reports, go anywhere from 7 to 45%. I mean, just think how many people in this room are suffering science fatigue right now. Approximately 1/5 of adults in 2016 were reported to have chronic widespread pain in the United States. And this was frequently associated with other symptoms such as depression, anxiety, fatigue, as well as headache, light sensitivity, all things that have been associated with symptomology in early Lyme disease. And then in another study, which is a little bit older but still relevant, over 80% of the general population may experience at least one somatic mutation within a 6-week period. And 81% of healthy university and hospital staff described at least one such symptom over a 3-day interval. So in the absence of an EM, it becomes very difficult to diagnose early disease based upon constitutional symptoms. So if not treated early, you can have dissemination through the lymph and the bloodstream. The Borrelia can disseminate to different regions of the body, notably the joints, the nervous system, and heart tissue, and you can have several manifestations of disseminated early infection. Some of this is multiple erythema migrans, where the bacteria returns to different areas of the skin and will form multiple erythema migrans rashes over the body. We've seen some people come in with hundreds of these. Seventh cranial nerve palsy, commonly referred to as Bell's palsy, joint swelling, radiculopathy, lymphocytic meningitis is fairly common in children, and more recently, it's been recognized that Lyme carditis is prevalent, which can even result in a full block and syncope. Importantly, following dissemination, the bacteria disseminate in low numbers in the bloodstream, and following dissemination, the bacteria clear from the bloodstream and are no longer circulating. Thus, it makes it very difficult to directly find the bacteria itself, because the timing, rate of dissemination, and ultimate site of dissemination will differ among different people. So it becomes very difficult to find the bacteria directly, and because of this, culture and PCR of blood has only been effective in about 20% of the cases. And then, if disease persists even further, you can have what are termed late-phase manifestations of Lyme disease. In the United States, the most common late-stage feature is Lyme arthritis, which is a monoarticular or oligoarticular large joint arthritis. It's usually associated with little or no pain and arrhythmia. In Europe, other manifestations, such as neurological manifestations or advanced skin manifestations, are more common. Late-stage disease is characterized by local inflammatory responses typically with no or few constitutional signs or symptoms. And then beyond this, there are individuals that have what is known as post-treatment Lyme disease syndrome, which I am not going to discuss today. So this is a chart that was recently released from the CDC, charting the relative frequency of some of the features of Lyme disease that were seen in a large number of patients collected between 2008 and 2017. And you see the majority of patients do have an erythema migrans, but other features such as meningitis, facial palsy, and arthritis are fairly common, although CDC admits that due to the constraints of reporting, late-stage symptoms may be over-reported in this kind of a setting. Typically, EM are going to be a bit higher than that. So in terms of diagnosis, it's important to note that most people, less than half of the people that have Lyme disease, remember being bitten by a tick. The erythema migrans is not always present, and it's not always recognized, but if present, is considered diagnostic, though atypical lesions do occur. In the absence of an EM or a recognizable EM, clinical diagnosis is very difficult. Some people would say it's impossible. And that direct detection of Borrelia burgdorferi by culture, PCR, or microscopy tends to be ineffective because the bacteria are present in low numbers and only transiently in the blood, and culture methods require specialized media and up to 2 to 4 weeks to grow. So PCR and culture of blood is only effective about 20% of the time. So what does that leave us with? The laboratory diagnosis of Lyme disease is currently based in indirect methods, specifically on the serological detection of antibody against B. burgdorferi in a 2-tier system. Typically, this is either whole cell sonogates or recombinant proteins from the bacteria. Now, this 2-tier system originally, up until very recently, consisted only of either an EIA or in older days an IFA. The IFA is rarely used anymore. And if this is positive or equivocal, you will perform a secondary Western blot. The Western blot would be if signs or symptoms were less than 30 days, you would perform an IgM blot and an IgG blot. If symptoms are greater than 30 days, you would perform an IgG Western blot only. This is due to a certain level of nonspecificity that's associated with the IgM blot. And specificity is the entire reason that a 2-tier system was put into place. It was to deal with nonspecificity that was associated with the initial EIA and or IFA tests. The Western blot added specificity to this, but it also introduced very low sensitivities for the detection of early disease. However, now, as of July 29th, FDA released new guidelines, which are supported by the CDC, saying that you can do a second different EIA as a second-tier test if your first-tier EIA is positive or equivocal. And this was driven by the approval of several new assays that occurred in the last 18 months or so. What I'm going to talk about mostly are the limitations associated with the standard second-tier assay, which was the EIA followed by the Western blot, as we're not really sure how the new paradigm is going to fully work. But the criteria for positivity for the Western blot was that if you're running an IgM blot, you had to have 2 of 3 bands positive, and over here you have depictions of an IgM Western blot and an IgG Western blot. And not just any 2 bands, it had to be 2 of these 3 bands. And then if you were looking for positivity in an IgG blot, you had to have 5 of 10 bands positive. What this means is that not only did you have to have a detectable antibody response, but that antibody response had to have enough diversity to be positive by the Western blot, which during early disease is problematic when the antibody response is still developing. Now, it's also important to note that these criteria and these Western blots were initially developed using cultured Borrelia burgdorferi. Now, as I mentioned, that there's a change in surface protein expression from the tick into the host and in the host during disease. There's also very different surface expression of antigens in cultured B. burgdorferi from in vivo expressed B. burgdorferi. So in the blots, it turned out later on that in vivo expressed antigens were poorly represented, which also affected sensitivity. Also, at the time the criteria were formulated, many of the antigens were not defined, and it turned out that a lot of these bands, like the 41 kilodalton band or the 58 kilodalton band, actually had several antigens in there. On top of all of this, the Western blot is typically read subjectively. So all of this combined to have a major effect on the sensitivity of these assays during early disease. So this was a conglomerate of several studies that were put together by Thiel in a 2016 manuscript that showed that for a first-tier EIA, you can have a sensitivity during early-stage disease of 74 or 75%. However, when you added the second-tier Western blot onto that, your sensitivity was reduced to less than half of that, 35%. So 35% sensitivity for a diagnostic assay for a disease like Lyme disease, not good. Now, as the disease progresses, naturally, you get better seropositivity because the antibody responses had more time to develop. But what it did add was it did add specificity. Now, the specificity in this particular study of the 2-tier assay was greater than 99%. It's a little higher than we've seen in other studies. Usually for 2 tiers, we see sensitivities between 35 and 50% with specificities greater than 97. Whereas the first-tier assays usually hover somewhere between 75 to 90% specificity alone. So why the importance of specificity? Well, the importance of specificity has entirely to do with positive predictive value and the pre-test likelihood of having a disease. So this is very important clinically when you're using an antibody-based test or any test for diagnosing a disease, or I should say, supporting the clinical diagnosis of a disease. So the predictive value of an antibody test is highly dependent upon the pretest probability of an infection in the patient being tested. So in a patient that could have Lyme disease, in the absence of an EM or with an atypical EM, the rate of constitutional symptoms observed in the general population is so much higher than the incidence of Lyme disease that unless your test is 100% specific, which no test ever is, it's more than likely that the person that's sitting in front of you with those constitutional symptoms does not have Lyme disease. Because of this, there's little value in using an antibody-based test to screen patients on the basis of nonspecific constitutional symptoms, unless you have a very strong suspicion of Lyme disease, such as you have a tick bite or the person has a distinct EM. Given this, even moderate reductions in specificity make the positive predictive value of a serological test for Lyme almost worthless. Thus, high specificity is key, and that's why the CDC and the FDA had stuck with the 2-tier system so long despite lack of sensitivity in early disease. So what causes non-specificity in the first-tier assays? Well, it's primarily use of whole cell sonicate, which is just broken up bacteria, or whole recombinant proteins. Why? Because many of these proteins have conserved regions that are similar to regions in other bacteria and have cross-reactive antibodies. Here's an example from Borrelia P41, which is the flagellin protein. A study back in 1991 showed that there were phylogenetically conserved regions of the protein, linear epitopes, that would bind to antibody raised against distinct bacteria. and had no relation to Borrelia whatsoever. It also had semi-converged regions that would bind to antibody that was raised against bacteria that were similar to Lyme disease. And, of course, it had variable regions where you could find areas that were distinct and had unique epitopes specific for Lyme disease. So we have been working from the hypothesis that we can use peptides that contain epitopes highly specific for Borrelia species to eliminate cross-reactivity and increase the specificity of first-tier assays while hopefully maintaining sensitivity and eliminate the need to have to do the second-tier test. So create a first-tier only test because the first-tier tests were of higher sensitivity. As proof of principle, there are 2 peptide-based assays currently in use, C6, which has actually been very successful, derived from the IR6 region of the VLSE protein, which probably doesn't mean anything if you don't work in Lyme disease, but VLSE is one of the major surface proteins of the bacteria and is involved in immune evasion, and has to be present because a lack of VLSE makes the bacteria ineffective at infection. And also there was PepC10, which has been used as in conjunction with other antigens in some assays. However, each of these peptides only contains a single epitope, which does limit efficacy somewhat due to antigenic variability among different species of the bacteria, as well as variability inherent to the human immune response. We've been trying to identify a minimum of 5 sensitive and specific epitopes that can be combined into a single assay, and that number comes from a statistical analysis done by Barber et al. That showed an assay with a minimum of 5 antigens would be optimal for sensitivity and specificity for detection of Lyme disease. So from all this, we get to epitope mapping. So we've used ProImmune's ProArray Ultra custom peptide microarrays to map a large number of different antigens, which are on the next slide. We used arrays that had 15 mers overlapping by 10 amino acids with a 5 amino acid offset. And we screened them with well-characterized early Lyme disease sera. So what does well-characterized mean? It means the patients met the CDC criteria for the diagnosis of Lyme disease. They all had erythema migrans that were distinct, greater than 5 centimeters. They had appropriate symptoms. They had exposure to a tick environment or had a known tick bite. And also, they were immunoreactive sera. We used patients that had highly immunoreactive sera. What does that mean? They had 9 to 10 clear bands on Western blot immunostrips. Using these sera and criteria saying that we were going to first evaluate only peptides that bound to antibody in more than 75% of the samples that we used for initial screening, we found over 80 peptides for further analysis. And this is a list of all of the peptides that we've mapped thus far. It's actually 22 because we did 2 OSPC types. And don't worry, I'm not going to go through all of these today, but I'm going to kind of give you an overview of how we've been evaluating the peptides once they've been identified. Sorry. So, OSPC, which is the type K type of OSPC. So OSPC is a variable protein that has several types. The epitope mapping revealed 3 peptides of interest, which we just generically named OSPC 1, 18, and 30. The first thing we did, and we do with all our peptides, is that we screened them using batches of sera from 10 to 20 immunoreactive patients from Lyme disease, and 10 to 20 normal sera collected in a region where Lyme disease is not endemic. In this case, it was Arizona. And in this case, we're just looking first to see for a separation between the Lyme disease patients and the normal sera, which we saw for OSPC 1 and OSPC 30, but not so much for OSPC 18, which actually the lines overlap completely. So why didn't we see reactivity here when we saw it on the first epitope mapping? Well, there we were using highly immunoreactive sera, and here we're using sera basically from patients that just had an EM without a prior knowledge as to their antibody levels, more real-world sera. So, and we would occasionally see this where some peptides just didn't react further and we didn't carry them forward. So we carried forward into our analysis the OSPC 1 and the OSPC 30. So the second thing that we needed to do was these epitopes need to be highly conserved among strains of Lyme disease-causing bacteria, because if there's too much variability, then as you evaluate people from different parts of the country, you're not going to have very good coverage or very good reactivity. And what we found is that aligning this with the sequences for a number of different bacteria that were present in the NCBI database, that OSC1 was actually very highly conserved with a few amino acid changes towards the C-terminus side of the peptide, whereas OSC30 was not. There was a lot more variability in the peptide. And we were comparing this to PepC10, which was one of the commercially available peptides that are already out there. So we moved forward with OSC1 and compared it to OSC10. And what we found is that when we moved to a large group of patient sera that consisted of people with erythema migrans positive Lyme disease, early Lyme disease, normal patients from a non-endemic region, patients with rheumatoid arthritis because they have an inflammatory disease with elevated antibody levels and joint destruction, RPR positive, these are people that had syphilis, which is caused by a spirochete that's related to Borrelia, or patients with late Lyme disease. We found that the OSC1 was actually efficacious at detecting patients that had early Lyme disease. We had a positivity rate of 62.1%. Positivity, equivocal, and negative, those were all calculated from antibody binding to normal control sera. Positive patients have a greater than 3 standard deviation, absorbance higher than normal controls, equivocal between 2 standard deviations and 3 standard deviations from normal, and negative are less than 2 standard deviations. So the OSC was able to bind positively to approximately 62% of the patients, which was actually better than the commercial peptide. So this was reassuring data. We did see some nonspecific antibody binding, particularly in the syphilis sera, although the assays that we're running here were basic ELISAs, so there was no optimization, and some of that we've been able to work out a little bit with different blocking buffers and so forth. So we began looking at some of the other peptides that were identified in the initial epitope mapping. So decorin binding proteins are 2 proteins, the decorin binding protein A and B, that are expressed by the bacteria and required for normal virulence. We found 4 peptides from DBPA and 3 from DBPB, all demonstrated on their own low to moderate antibody binding. However, and is here showing the 2 best singular peptides, one from DBPA, which bound to approximately 40% of the patients, and DBPB, 48%. However, when we mix them together, they actually work better than any other peptide combination that we've seen to date, where they were able to positively identify 87.5% of patients, another 6% equivocal, which means only 6% of the patients here that we used for screening were negative. We did still see some non-specificity here. And again, we've been working on that using different methods. What was also interesting is that when we combined the 2 peptides together, they actually worked better than the 2 parent proteins that the peptides were derived from when they were mixed together in an assay, which was very interesting. Now, the only drawback, it turns out, to using these peptides is, again, variability among different strains of bacteria. In this case, not different strains, but different species. There was very good conservation of the peptide sequences. Here I'm showing peptides for DBPB, but we have similar results for DBPA within species. So all Borrelia burgdorferi found in the United States seem to be very well conserved for these. But if you compared it to Borrelia guerinii or Borrelia afzelii, you saw a lot of amino acid sequence variability, meaning that if you were to use this in a diagnostic, it would be limited to specific locations, and that if you were to use the test in a different location, you have to derive other peptides from these 2 antigens, which reduced the enthusiasm for these particular peptides for diagnostic companies. As I had mentioned previously, we expected that there was going to be a high rate of cross-reactivity for some peptides. And when we did our initial antigen screening, we only used Lyme disease positive sera. We didn't use normal control sera. In fact, if we didn't find any cross-reactive epitopes, I thought that would be a little suspicious. But for this protein here, which is BBK32, which is an adhesin for Borrelia and is also a complement binding protein, we saw that we had 1 peptide that had some moderate binding activity. I didn't put the numbers up here, but it was about 45%, not terribly great. But we had another peptide that had what I called universal cross-reactivity, you could not distinguish the Lyme disease sera from any other sera that we used with this, which was actually quite striking. And we later found out through sequence analysis that this particular peptide had a high degree of homology to a peptide in common food proteins, including corn, rice, millet, and other grains. And we think that might be a source of actual cross-reactivity here. And this was something we also saw in another protein, which was P66, which was another adhesin. It produced only cross-reactive linear epitopes. We actually found 7 or 8 epitopes from this one peptide protein, which is quite large, but all of them kind of produced this kind of format, which was quite interesting. But the last one I'm going to discuss today is the one that's actually ended up in the test that's coming out in the market. We had previously developed, actually before I joined the lab, Dr. Datweiler had developed an optimized version of the C6 peptide sequence. It was optimized by modifying the sequence to generate more of a consensus sequence that better represented diversity that was observed in different strains of Borrelia and from different regions. We had been working with this because it was fairly immunoreactive on its own, but it increased the immunoreactivity of other peptides when we bound it to it. So we came down to 3 that worked very well with it over the course of our screening. A peptide from P35, which is a protein expressed early of unknown function, ERP, which is a complement binding protein, and a peptide from Flagella. These peptides alone had moderate binding ability. However, when we linked them to this VLSE epitope, they were much, much better at identifying patients with Lyme disease, moving from 16% to 56%. And the FLA peptide was even better, working at 62%. And then with collaborators at Bio-Rad, we took these peptides and we attached them to beads in their bioplex multiplex assay. And this further increased the sensitivity of the peptides. So this FLA modified VLSE was able to identify 80% of patients in one cohort, and these were all, this cohort number 2 was from patients that were diagnosed with Lyme disease and positive via the IgM or IgG Western blot. So in one respect, you're sort of stacking the deck there because they're already serologically positive. The other 2 detected 72%, and when you used all 3 peptides together, you detected 81%. But the important thing was that all of these were done with a specificity of greater than 99% in the assay. And then taking a separate cohort where you just took people where a physician said, you have Lyme disease, we were still able to, and you know the 2-tier analysis wasn't up here, but it was far less than 58%. We're still able to detect disease in 58% of the sera. So in the multiplex format, the peptide performance was even improved compared to the ELISA, and we're happy to say that this antigen was part of the antigen set that Bio-Rad included in their total Lyme multiplex assay. So where are we going from here? So in our current and future directions, we're continuing to move forward with evaluating peptides that we've identified in the epitope mapping, because there was a lot of them. But we're also trying to move towards evaluating peptides in a POC format for use in office. This is kind of one of the main areas that diagnostics are going into today because everybody wants an answer within 5 minutes. We've had some we've had some challenges in adhering peptides to membranes, but we're working through that. Different peptides seem to require different anchors, and it also depends upon the membrane. We're also evaluating the use of peptides as antigen targets in a completely different type of assay for Lyme disease. We're trying to develop a quantiferin-like assay for stimulating T cells in an interferon gamma release-like assay for Lyme disease. And then a third thing that has just recently come up is now it's been recognized that there's cross-reactivity for both the C6 peptide as well as other antigens with relapsing fever Borrelia, which is they're phylogenetically related bacteria, all in the same genus, but these cause a different disease. And they cause a different disease in different areas of the country. And what we're seeing is rates as high as 4% positivity in Lyme disease tests in regions where Lyme disease is not present. So as a result, our lab and others have begun investigating modified peptides to try to distinguish these 2 diseases and possibly eliminate further some of the background reactivity that's seen in Lyme disease tests. So thank you for listening, and I'd just like to acknowledge my compatriot and mentor, Dr. Raymond Datweiler, who started all of this in his company Biopeptides Corp., our former technician Maria, and present and past students. Maria Gomes-Selecki was a previous member of Biopeptides, who's now in the University of Tennessee, as well as collaborators at BioRad Labs. And this work was all funded by the NIAID SBIR program. So thank you, and I'll take any questions.
Thank you, very interesting approach. So I have two questions. Every time that I look at antibody binding to peptides, obviously my mind starts to wander. So the two questions are, number one, is it possible that the binding of an antibody, that's more of a theoretical question, right? So the binding of an antibody to the linear peptide is somewhat different from the binding of the same, the possibility of binding to the peptide once embedded in the protein, because they have a different degree of freedom overall. So the entropy changes. And so some of the differences you see when you actually have the peptide bound to the beads of the luminex is different than have it in the ELISA, because at least you have at the anchor on one side, right? So if you could comment on that.
So yes, that is something we see quite often, that the matrix that the peptides are being used on, what they're being bound to, does affect the reactivity. Certain peptides work better on the beads than they do in the plates. Others work better in the plates than on the beads. Everything seems to work better on anything but membranes. It's been, so it is kind of hit or miss trying to figure out how the peptides are going to work in certain situations. And we haven't really seen any specific structure like saying, well, this peptide is hydrophobic and it works better here, or this very hydrophilic and it works better here, or it's more acidic or basic. But, you know, I'm not a chemist, and there may be certain aspects of that that I don't know or understand.
Okay. And my second question is, so obviously there are also, three-dimensional epitopes.
Yes.
That could be of interest, right? But these are not the one targeted by your approach, because these are the linear ones.
Yes.
So is it possible that you're going to incur in false negatives, because there is a chance that you're going to have, randomly a greater response against the three-dimensional epitope rather than the linear, but you're looking only at the linear ones, and therefore you might be missing some of that?
Yes, there is a possibility in that, which is why we're trying to include, at least a minimum number of antigens to try to mitigate that as best as possible. But there is definitely. And you had mentioned before about differences in the peptide binding versus when they're in the protein. Actually, with the C6, which has been very successful, one of the interesting things is that peptide, when it's in the protein, is completely hidden. It's internal.
Right.
So it's good diagnostically, but if you wanted to try to use that, in some kind of protective fashion or something like that, it's actually useless.
Okay.
So
thank you.
I had the same question, but I'll continue on it. And I think the only thing I would like to know is that if you did take the people who did respond in the peptide or showed a positive reaction, would you see that their T cells would have a memory to that specific epitope? And would you be able to prove that yes, maybe there are coexisting epitopes? Because whenever I think antibody, I think of a conformational epitope, unless these epitopes are able to bind the B cell in a way that it is still functional, and you can see that. So is there a way for you to prove, or has someone done that study to show that maybe the T and the B cell epitopes are coexisting?
So we have seen some overlap in B's. We've also been doing some T cell epitope mapping. So we have seen some overlap in some peptides where some are also recognized by T cells, but not many. And I think it's more of a kind of a linked recognition thing, so you can get class switching to specific epitopes from B cells that aren't from T cells that aren't necessarily specific for the same epitope. In fact, they shouldn't be specific for the same epitope. But but we do see we do see that there are positive reactions to whole antigens. So if we have B cell, if we have a antibody that binds to an epitope from P35, then there's also some reactivity in that patient to P35 antigens, maybe not the same antigens, but antigens, T cells as
Okay.
well. Yeah.
All right, yeah, so I think that answers my question, and
Mhm.
I think the other one is more about if you are doing a longitudinal look at a subject from early to late progression of Lyme disease, and you have these specific reactivity which comes up, do you see a change in the affinities of these antibodies towards the same peptide?
So we haven't done really affinity-based assays. We just look for basic reactivity. And yes, usually when a patient comes in the acute stage at their first visit, if they're positive for a particular epitope, they remain positive. What's interesting, though, is that we found that if a person comes in most of the time in the acute stage, if they are not positive for an epitope, they typically don't become positive. Now, there's been this whole consensus in Lyme disease that usually if you don't see a response early, you look for seroconversion. But a lot of times we don't see seroconversion. You know, either they're positive at the first visit and they remain positive, or they're not positive and they never become positive. Now, that may have a lot to do with when they're treated, what the stages of the immune response, how long they've had the bacteria, so forth. But that's what we've observed so far, and I know others have observed the same thing. Okay, thank you very much.